So I started talking about these before. These are the actual checkpoints of the cell cycle. The first of which is found in the G1 phase, the gap 1 phase. And this is the gap 1S checkpoint, or the start or restriction checkpoint. Just synonyms referring to the same checkpoint, which happens right here. Okay, and again, this is the checkpoint in which we are able to determine whether the cell is capable of actually moving into the S phase. So if the cell has all the proper components, if it's the right size, to be able to actually undergo DNA replication during the S phase. The second checkpoint we have here, and the G2M checkpoint, okay, which happens towards the end of the G2 phase, and what this checks for is proper DNA replication, okay, and we also check to make sure that the cell is the proper size, and that we have the proper components to undergo cellular division. From there, we have the spindle checkpoint, which happens basically within the phases of metaphase and anaphase, where we have the cell checking to make sure that all the spindle fibers have connected properly onto the kinetic course, okay, before we actually separate our chromosomes during the process of anaphase. Okay, if something essentially goes wrong with this checkpoint, we end up with a non-disjunction event, which we talk about a lot more in the next week's workshop. Okay, and these non-disjunction events lead to diseases like trisomy 21 Down syndrome, amongst other genetic diseases that are related to aneuploidies. So these checkpoints are controlled